prediction algorithms, and Jim Costello will give the talk.
First, thanks for organizers for inviting me. And okay, this is the last talk of the day, so I will make it as exciting as possible. So what I'm going to be talking today about is really a dream challenge, and the results of the dream challenge, some insights we gained from that. And this has all been published very recently in Nature Biotechnology and at the Advanced Online publication. I'm the first author on the paper, so please look that up. There's going to be much more information associated with the algorithms and things in the actual paper itself. So first of all, I hope that most of you are at least somewhat aware of DREAM or kind of know what it is, but to kind of make sure we're all on the same page here, it stands for the Dialogue for Reverse Engineering Assessments and Methods. It's a community effort to really kind of bring together as many people as possible to focus on particular challenges we all have and to kind of nucleate around data sets and questions and try to address systems biology and translational biomedicine types of questions. So how these things come about, we usually have either a group of people or a group of data that we have interesting questions for. We want to make sure that the data is not published because if you guys have access to it, then you can use it and it wouldn't be a completely unbiased assessment. So once we have the data and we have a good question, we want to make sure it's nice and clean and available for you guys to use. We supply it to you guys along with the question. And that really represents kind of the open challenge period. From there, we invite anybody who's interested in participating to come in and, apply their most favorite algorithm, their best approach, some crazy stuff they can think of to see if they can solve the problem. We've had some interesting applications here where people have built like little games and avatars where people, you can play your little avatar and try to solve mutations and so on and so forth. Once you're done with that, then we actually evaluate the methods against, again, a held out, unseen data set. And, we want to make sure we have really good, rigorous scoring methods because this could potentially become a large benchmark data set that could be used for future algorithm development. And people use this quite often as far as developing new methods and reporting in grants and papers. So how to make this successful? So we want to make sure we have an interesting and clear question, something that the community is very interested in. Again, the data has to be unpublished, so we need to make sure that we can actually properly benchmark. Incentives. So we hope that most people participate in these challenges because they're very interested in the biology and the question and developing the methods, but there's also some tangible benefits to doing it. So, for example, I'm talking about a Nature Biotech paper where the top winners were invited to be co-authors, so it's a nice publication for them. Along with this particular challenge, since we participated with since we partnered with the NCI, the top two teams were given $100,000 grants to improve their methods. So there could be very tangible results to winning some of these challenges. Organization, there's a ton of people behind this, and I'll talk about them at the end, but know that this is a lot of work. Open lines of communication. So we want to make sure that we're really, the effort here is to kind of bring people together and help us nucleate around data and questions. So we want to make sure we're as transparent and open as possible. And then making sure that the scoring is very rigorous, it's well supported, well justified, and could potentially be used as the benchmarking standard for the community. And this is being done currently with somatic mutation calling with the ICGC, and there's a challenge currently going on where the winner of that challenge is actually going to be the standard method that's going to be applied to all ICGC data. So again, these things have very tangible results. So I wanted to give a quick note on the recent kind of partnership between Sage Bionetworks and DREAM. So Sage was founded by Steve and Friend a couple years ago with, again, the initiative to really push open science and try to get people to collaborate, share data, work together, so on and so forth. And one of the systems that they built is called Synapse. This is just kind of an infrastructure type thing where you can work together, share data, download data. There's an R package that you can download. I would check it out because there's some really nice, well annotated data sets in there. So this kind of represents some of the challenges that have been run in the past. Is there a pointer here? Oh, yeah, okay. And DREAM was founded in about 2006. 2012 is when Sage partnered with DREAM. And from there on out, everything's going to be run through the Synapse platform. So all the data is going to be there. All the algorithms that people use are going to be there. All the insights and stuff are going to be located there. So it's a real nice resource for algorithm development. Okay, so what I'm really going to talk to you today about is the NCI-DREAM Drug Sensitivity Prediction Challenge. This is set up to basically be, can we take multiple omics data sets and use that information to then predict drug sensitivities? So it was designed using a panel of 53 breast cancer cell lines, and the data was donated from the Joe Gray's lab at OHSU. They screened a whole bunch of genomics, omics profiling technologies, along with doing drug response on a whole bunch of different drugs. There were about 30 drugs that were unpublished at the time of running the challenge, so that's why there's 30 drugs in this. Again, sampled across all of the cell lines, so we supply the community with the genomics profiling data sets, the training data set, which is 35 cell line responses, and then they asked the community to predict for 18 cell lines what their drug response is going to be. You know, people go out and apply their favorite methods. We had 44 total submissions with this challenge, and then we, score, and I'll talk about this stuff and some of the insights that we gained from this challenge. I'm going to mention this only briefly, but there was also two challenges within this group of challenges, and this is on drug synergy. This particular challenge looked at profiling of about 14 compounds over different time points and different concentrations, along with some copy number variation data, and again did drug response profiling for those for one particular cell line. And the challenge was, can I take all single profiling data sets and predict what the combinations are going to be? Are there going to be synergistic interactions, antagonistic interactions, so on and so forth? And I'm not going to talk about this, but just only to mention that we should hopefully hear very soon if this has also been accepted to Nature Biotech to be published as companion papers together. Okay, so for the challenge, for the NCI Drug Sensitivity Challenge, the data that we actually have that we supply to the community is copy number variation data, RPPA protein quantification, gene expression microarrays, methylation, RNA-seq, and then exome sequencing. Along with that, again, is the drug response measurements. So this is a typical 72-hour drug response assay. Treat cells for 72 hours. At the end, you do your typical cell titer glow responses. You have your dose response curve. You can calculate your various metrics from that, including GI50, which is the metric we used. And then you have basically for any particular drug, in this case an AKT inhibitor, you have a kind of a range of response across those different cell lines. Thing to note too is that I'm going to be reporting everything in negative log 10 GI50 values. So the higher the number, the more sensitive the cell line, okay? So this is just meant to demonstrate that there's a range of values you're getting, response values you're getting for individual drugs, and then even within drugs you get a range of response, just showing that you get nice diversity in response overall. So again, we asked the community to take this data and predict how a panel of 18 held out cell lines would respond to drug. So in that, we need to talk a little bit about how we actually scored a particular team submission. And in this case, we used 3 different approaches, arrived at kind of using this top one, the weighted probabilistic concordance index. But it is important because, again, we want to make sure that these are, nice, well-established benchmark data sets that can be used in the future. So the concordance index, which most of you should know, is a relatively simple measure that just asked if how concordant an observed list is to a predicted list. And in this case, all you're doing is testing all pairs. You're saying is A and B ranked the same in the predicted as it is in the observed? So A is ranked before B in the observed. It's also ranked before B in the predicted, so it's concordant. If B is ranked before C in the observed, but C is ranked before B in the predicted, then it's not concordant, right? You do this for all pairs, and you come up with a metric that basically represents how concordant these two lists are. The thing that we have to account for is that you do have variation in the individual drug responses. So we want to be able to account for that variation in the scoring metric itself. So we can directly incorporate that into the metric, and that's why we call it the probabilistic concordance index. And then the reason we call it weighted is because we weight this across all 28 drugs that we used, okay? So the scoring, becomes one score across all 28 drugs. The other scoring metric we used was the resampled Spearman correlation. And in this case, what we're doing is we're actually creating an ensemble of gold standard ranked lists sampled directly from the data itself. So in this case, you could actually get, for two cell lines B and C, since they have relatively the same response, the same distribution of variation, you could interchangeably, rank these, 2 or 3. So we want to be able to account for that. So we take this ensemble of basically gold standard rankings, compare them to the predicted, we'll get a distribution, compare that to the null model, and see if we're getting a difference in separation and performance. When we compare these two metrics, we have the WPC index here. We have the teams ranked according to the resampled Spearman. You see that you get a nice correlation in the two metrics. There is some variation in there, but the thing to notice is that the top 2 actually are the top 2, and they're consistent. The other method we looked at was to look at balanced accuracy. So the previous methods were looking to actually score based on the full ranking itself. This is actually looking to say, okay, can I predict sensitive or resistant just as general kind of binary categories, where the middle stuff is ambiguous. I can't really make a call, so I'm going to kind of ignore that. So we can do clustering to identify what's going to be sensitive or resistant. We do this across all the different cell lines, across all the different drugs, across all the different cell lines. And something like the gold standard would look like this, where the red is the sensitive, the blue is the resistance, and they're all ranked accordingly. The actual results of team predictions look something like this, where you have, this is the top performing method, and this is the least best performing method. We don't say anybody's the worst in DREAM challenges. So in this case, you can see that there's more red at the top, which is what you'd expect. There's more red at the bottom here, which is showing that there's not as good a performance, but this is just kind of another metric we use to evaluate the teams. When we compare the balanced accuracy to the WPC index, this is not ranked, this is the actual values themselves. So we're actually looking up in this corner for the top performing teams. Again, the top 2 performing teams are the top 2, although they do switch. So you do get different information from different metrics. This represents the kind of distribution of all team performances. What we did is broke them down according to our best guess as to kind of what the underlying methodology was. So things like ridge regression, elastic nets, lasso, these types of regressions are all going to fall in the sparse linear category. Things like random forests are going to fall in nonlinear, and so on and so forth. So these are the large categories, which kind of gives you a sense of how people kind of attempt to solve this particular type of challenge. The other thing we did is we did a kind of a robustness scoring analysis where we did a resampling of the gold standard. So we mask a certain portion of the gold standard, we rescore, we mask, we rescore, and so on and so forth, which then creates a distribution of scores for any particular team. And that's how we determine if the top performing team is in fact the top performing team, or if it's not just an artifact of the data. So what you can see is at least the top 2 teams are really robustly ranked 1 and 2. It gets a little more blurry in 3 to 14, and then it kind of tailors out from there. So the top performing team is a team from Finland, and this was led by Sami Kaski. And again, to emphasize the implications of this, they're invited to give talks at the DREAM conference. And after Mehmet got down from talking, because he presented the actual methodology himself, the people at Sage Bionetworks were so impressed they offered him a job on the spot. And he was hired by Sage Bionetworks and now is with Adam Margolin at OHSU. So what did they do? So this is the actual kind of summary of the methodology. And I'm going to summarize it very quickly, and I want to summarize the second best team very quickly. And all the details are contained within the actual paper itself. So the first thing that they did, which I thought was interesting, is they didn't use just the data as it was given to them. They actually projected that out into other forms of data. So what they did is what they call views. And for example, the gene set view is just you project your, for example, gene expression data into a gene set space, and you calculate the putative gene expression, pathway expression or gene set expression. Combination views are they actually combine things like methylation and gene expression to see if they have any relationship. And discretized views is pretty self-explanatory. They just discretize the data to take it out of a continuous space. The next thing they did is instead of just taking the features and trying to regress that against the response variables, which in this case are the GI50 values, they actually did they applied a kernel function here, which was either a Gaussian function for the continuous or Jacquard similarity measure for the discretized values. And then they calculated n by n or a cell line by cell line, what they call kernel matrix, okay? Simultaneously, what they do is they calculate the weights of these kernel matrices based on, standard machine learning, leave one out, cross-validation types of approaches. And they also looked at the GDSC, the Sanger set, kind of similar to the CCLE data set. So they learn the kernel weights, they learn the kernels, they do kind of a weighted summarization to have their final kind of kernel matrix. Simultaneously, they learn the individual cell line weights according to the drugs, using the same type of approach, and also including the GDSC data. You combine the kind of the kernel matrix here with the drug weights, and those end up becoming your predictions. The second best performing team was from Texas Tech, Quinn Wan and Ranadip Pal. They used a different approach, but they used the same principle in trying to model nonlinearities in the data, which is kind of a take-home from the paper. If you're able to model that to account for the nonlinearities in the data, you can actually gain performance. So in this case, they took each of the individual data sets, the 6 data sets sampled. They create regression forests from each of those data sets, and then they create kind of an ensemble of combined methods, so the power set of all random forests, weight those accordingly, and then do an evaluation on leave one out, cross-validation to select the best method, apply that best method, and that's their final integrated model. And again, you can find much more information on their method. They published a separate paper in PLOS One. Okay? So some observations from the individual method performance. For the majority of people, 23 out of the 44 used all 6 profiling data sets, but some used less, though we couldn't find much of a performance difference if people used all 6 versus 1 or 2. Okay. Outside information was the one thing that we actually found that actually contributed to overall performance. So if they were able to incorporate biological pathway information, or they used outside information like CCLE to help train their models, they were able to, on average across these methods, gain in performance. Again, I mentioned nonlinearity, so if you compare the sparse linear regression versus the nonlinear regression type of approaches, you do see that you definitely gain performance, overall performance, by being able to try to account for those nonlinearities. And the top 2 methods were able to do that. The sparse linear methods, elastic nets, and so on and so forth, while they're great methods, they do miss certain things. And being a dream challenge, you always have to test if the wisdom of the crowds phenomenon does hold up. Of course it does. So if you integrate multiple independent pieces of information, you tend to gain in performance. We can also look at, so we just looked at kind of observations that were made from individual methods. We also want to look to see if we can gain some insights regarding individual drug predictability, or how informative the individual data sets are in their predictability overall. So in this case, what I'm showing is basically this is the full set of predictions, ranked ordered according to team, and then shown according to drug. And these little weights at the top represent kind of how much information, so to speak, is within each of these individual drugs. So for example, chloroquine, which I know is down here, was very poorly predicted, and we didn't weight that very highly because there was issues with the actual measures itself. What I'm showing here is kind of the relative distribution, or the actual distribution, of individual drug performances. So this is all 44 teams, distribution of all 44 teams. Little red dots represent the gold standards. And what you can take away from this is there's still much room for improvement, so we can still try to get closer to those red dots. But for the most part, almost all the drugs, except for the last couple ones, had some predictive signal in it. And overall, we're able to actually make reasonable predictions, at least better than a random model, which would be a 0.5, PC index of 0.5. Okay? So the other thing we can do is we can kind of group these pieces of data and look at an individual method and see how the data sets themselves contributed to the predictability of that drug. And in this case, what you can see is, at least in the Bayesian, so the Bayesian multitasking MKL was the top performing method, and we worked with them to generate some of these additional data sets. We also implemented an elastic net ourselves, just a standard kind of off-the-shelf GLM net from our package implementation of the elastic net. And this, there we go. And the only thing I wanted to point out here was, the question was really, are there drug classes, are there subsets of drugs that tend to be more easily predicted, or could use can be more easily predicted with expression versus exome sequencing or things like that. And for the elastic nets, we didn't find a whole lot of good information, but at least for the Bayesian multitask MKL one, you can see that this kind of group is a little more red, meaning better predicted, than some of the other groups. And you can kind of see this as a summarization here. And at least the Bayesian multitask MKL method was able to kind of leverage the gene expression data a little bit more than some of the other data sets. So again, we wanted to kind of evaluate how these individual data sets contribute overall. We ranked these according to performance of ElasticNet. You can see that RPPA was the top performing, though not statistically different than the expression data. And this is likely due to the fact that RPPA just has many less features. So even though elastic nets are supposed to be a way of filtering out the non-informative data, if you pre-filter that data before you throw it into an elastic net, it'll still improve performance if you're selecting the proper features. So RPPA only has around 200-ish data points, so that actually contributes significantly to the ability of the predictor to predict. The Bayesian MKL method was able to use gene expression data from microarrays as the most informative piece of data, and this was pretty consistent amongst all of the individual teams. So we asked them what was the most informative piece of data that you guys, if you had to pick one, what would you pick? And people generally picked gene expression data, microarrays. And I think that's largely due to the fact that we're very well versed in microarray analysis and prediction methods and so on and so forth. Although RNA-seq was not performing very well overall, interestingly. Then we can look at these additional data views, which I'm not going to talk too much about, only the fact that you can get a little bit of gain in performance if you incorporate some additional information. You project a particular data set into a different space. So if you look at pathways, for example, in gene expression, you can gain a bit of performance. And then the other thing to note is that, at least for the Bayesian MKL method, if you take all the views together, you can get the best performance. Okay, some observations from the drug and data set component of this analysis. So it appears that the growth signaling drugs tended to be a little more easily predictable overall. When we look across all the data and we look across all the methods, gene expression microarrays were the most informative single piece of data. By calculating different views, you can actually gain information, which is pretty interesting when you think about, this becomes more applicable in the clinic. When you think about a clinical assay, you're not going to be measuring RPPA, gene expression, methylation in the clinic. If you had to measure one piece of data and you're actually able to amplify the informativeness of that data, that's a good thing. So this is kind of what this is showing. And then the complementarity was different amongst different methods, at least in the Bayesian MKL method. Microarrays combined with exome sequencing gave you the best performance. I apologize, this is supposed to be microarrays and RPPA data here for the elastic nets. So with that, I'm going to talk about some of the people I referred to earlier. There's a ton of people involved in this. These are some of the organizers. So Joe Gray and Laura Heiser were great in kind of putting the data together. Gustavo is kind of the mastermind behind the whole DREAM thing. Julio and Michael were great in helping with the scoring. Mukesh is really running the second part of the challenge, the synergy challenge, but was also helpful in scoring. Jim Collins, my old advisor, and Dan Gallahan and Dina Singer at the NCI for their support both through the challenge and also financially too. This is the community, so there were really over 100 people that participated in the challenge and really helped put this together and helped made this work and kind of helped us come up with these insights. And then this is just a little plug for the currently running challenges that were just released recently. There's one on Alzheimer's disease prediction, AML prediction, and then there's one about gene essentiality. So if you guys are interested, please check out the URL about that. And with that, I'll take questions if there's time.
Hi, nice talk. I have a kind of provocative question, but my own experience with students and colleagues is that sometimes when you tell your students, to solve a problem with a method, like use a elastic net on this data and make a prediction, you get different predictions just because you have, many many ways to use the method.
That's
so
right.
my question is, here you compare the teams in terms of methods, obviously, but from your opinion, what's the most important? Is it the method use or is it the team? Like comparing different dream challenges, you observe that some, I don't know, some teams always
Yes.
perform well, whatever the method, and the opposite way, or this kind of stuff?
Yeah, so we consistently see through these challenges that it's not the underlying method that makes the biggest difference. It's the team-specific effect, or it's the filtering of the data. So feature selection, obviously, is a very big factor. And if you're able to effectively do feature selection, you can throw it in just about any method, and it's going to be pretty good. Any other questions?
So this wisdom of crowds model is extremely productive. I'm thinking if there is a computational simulation, if you can run of trying, like within the same group, try to generate multiple predictors on our own and build an ensemble and then say, you know, are we really getting wisdom of the crowds?
Sure.
Or are we getting the wisdom of the data or the methods or whatever?
Right, right. So interestingly, we did something similar to that in the network inference challenge, where people did submit an ensemble predictor, and there were ensemble predictors in this challenge also. And even though a team runs an ensemble approach and submits the ensemble approach, still combining that with other groups' predictions still increases performance. So it's a weird phenomenon, but yeah, you still gain performance by having completely independent groups, even if they're implementing the same method, like you were suggesting, have different overall performances, and you can kind of leverage those against each other. So it does appear to be a group independence effect rather than the method independence effect.
Since you mentioned this before, does that relate to the feature selections that the individual groups made? Do groups that select non-overlapping feature sets, are they more complementary?
Yeah, so that's a tough one to answer, unfortunately, with the design of the challenge, because you know, people submit their solutions, and you know, they sometimes don't tell us what features they use. We were able to kind of do a sub-sampling with some of the, you know, the people that were willing to work with us on this, and you do get complementarity in, I mean, there's a high overlap with the most predictive features, but the stuff on the border that actually can give you the slight gain in performance is different between the different teams. Yeah, go ahead.
So given that this is more of a political question rather than a
Sure,
technical
okay.
question.
Yeah.
You invited the top 2 teams
Yeah.
to co-author the Nature Biotech article with you guys.
It was the top, so the deal was the top 1 team actually was co-authors, but the second team was mentioned directly in the paper and referred to, but yeah.
Okay, so but you must be coming across this situation in pretty much every challenge, right? So
Sure.
how do you guys address that?
As far as who,
Yeah, I mean, who to invite?
so I mean, it is based off of a ranked performance. So, you know, we can determine who is the best performing team or method. So in that case, it's pretty easy. The tricky ones is when you have methods that are not statistically separable, right? So if 2 teams are performing roughly close to each other, then, you know, typically we'll invite them both to be authors on the paper. We try to be as fair as we can with that. And then everybody is, so the NCI-DREAM community itself is considered an author on the paper. So all those people that participated are, you know, if you search PubMed, it'll come up with the publication. So everybody's included. It's just the top performing is actually is in the main author list. So, yeah. Mm-hmm. That's right. Yes, so I didn't put Chad up here, but Chad Crichton was the 3rd best performing team, although he's an individual. And he did a very simple thing. He just did linear regression with the features and the responses. And he ranked 3rd, though statistically he was not different from, it was 3 to 14 were roughly the same. So the top 2 methods were in fact the top 2 methods, and then 3, again, it gets a little blurry. I mean, by definition, we're interested in the best performing method. So whether it's simple or complex doesn't weigh into how we score, though you might personally think that a simpler method would be a better method.